Do you know
Oxygen can turn off
and on switches like this? But I'm not talking about these switches, but the switches of our genes. Switches in our genes. Are you kidding me? No, I'm not. And really, switches, because our bodies have to make every protein in just the right amount. For example, the hormone erythropoietin, which tells our bone marrow to make red blood cells, and red blood cells carry oxygen to our cells. So, for example, when our cells already have enough oxygen, they will make very small amounts of this hormone. But when they face a shortage of oxygen, they will start making this hormone in larger amounts. But how do our cells activate the switch of the gene that makes this hormone in low oxygen levels? Let's see how.
There's this protein called hypoxia-inducible factor 1 alpha or HIF1 alpha in our cells. When oxygen levels are normal, the enzyme proline hydroxylase happily adds 2 hydroxyls to most of the HIF1 alpha molecules. Next up, a protein called VHL or von Hippel-Lindau comes and attaches small proteins called ubiquitins to the hydroxylated HIF1 alpha with the help of enzymes. Ubiquitins are like tags that tell organelles called proteasomes to break down the HIF1 alpha and recycle the ubiquitins for further use. But when oxygen levels are low, most of the HIF1 alpha remains unhydroxylated, so no VHL binding and no ubiquitin addition. And the HIF1 alpha survives! Inside the nucleus, we can see the EPO gene and next to it, a DNA region called HRE hypoxia responsive elements. HIF1 alpha binds to a protein called ARNT and together they form the HIF protein complex. It sits on HRE and activates the EPO gene, meaning it can now transfer the protein making recipe to an mRNA which will go outside the nucleus and start making the EPO hormone.
We can imagine it like this.
Let's say HIF1 alpha has only one path and it leads it to proteasomes. There is this other path but it's blocked by two obstacles, oxygen and VHL. So HIF1 alpha is degraded when both of these obstacles are there. But if either of them is absent, HIF1 alpha can easily take the other way around and join with ARNT to form the HIF protein complex.
The von Hippel-Lindau or VHL protein can be absent when the gene that makes it is somehow mutated. And it happens in the von Hippel-Lindau disease, when people suffering from this disease are more likely to have tumor and cancer. Just think about it, if the HIF1 alpha survives because the VHL is absent, the cancer cells will easily be able to manage more oxygen for themselves, right? We talked about switches, remember?
We can compare the bulb to the EPO gene and the switch area to HRE. When the HIF protein complex comes and pushes the switch, it kind of closes the circuit and like this bulb, the EPO gene is turned on.
So that's about it. Now I have a question for you. Can you guess how people living on mountains have higher red blood cell counts?